Hello everyone. Today's topic is Lac Operon. We all know that human beings are made up of cells, and these cells contain the genetic material, which is DNA, and the DNA is responsible for the growth and the development of any living organism, including human beings. So, some genes will be required to be expressed all the time. For example, if we take genes which is responsible for the vital process which is respiration, these genes has to be expressed all the time because we respire all the time. So it is necessary the genes which brings about this process has to be expressed all the time. Whereas other genes are not expressed all the time, they are switched on or off whenever it is needed or required. Before beginning, we need to understand what operons are. An operon is a group of genes. These genes will be transcribed into mRNA at the same time. What is the role of an operon? The role of the operon is to control important biochemical processes. As already we've seen, respiration is a biochemical process which has to be expressed all the time. So, these operons are very important in prokaryotes to perform or to express the genes into proteins or enzymes. We have a question whether these operons are present in human beings or prokaryotes. The operons are not present in human beings, they are present only in the prokaryotes. What is Lac Operon? Lac Operon is a polycistronic gene, which means poly refers to many, cistron is functional. So this lac operon is a lactose operon, a polycistronic structural gene which is regulated by a common promoter and a regulatory genes. Who discovered this lac operon concept? So the picture you can see Jacob Monod and Wolff. So Francois Jacob and Jacques Monod were the first to elucidate a transcriptionally regulated system. This lac operon are usually present in bacteria such as E. coli. So the lac operon consists of three genes, and these three genes are involved in processing the sugar lactose, which is a disaccharide. There are three genes, which is Z, Y, and A. One of the gene is LAC-Z, which codes for the enzyme beta galactosidase. So, what's the role of the beta galactosidase? Is this enzyme hydrolyzes lactose into glucose and galactose. Since lactose is a disaccharide, it has to be broken down into glucose and galactose. The enzyme which is responsible for this action is beta galactosidase. Lac operon is an inducer operon. So the presence of a chemical switch on the operon is in the inducible operon concept. So the lac operon consists of one regulatory gene, which is I gene. Here you can find the I gene. So this I gene is the regulatory gene. Here I doesn't refer to an inducer, but it is derived from the word inhibitor, and there are three structural genes, which is Z, Y, and A. So the I gene codes for the repressor of the lac operon. It codes for repressor, whereas the Z gene codes for the beta galactosidase, which we otherwise call it as beta gal, which is responsible for the hydrolysis of the disaccharide, which means it breaks down the lactose into glucose and galactose. And there is another structural gene, Y. This codes for the enzyme permease, which means the lactose that is produced outside has to be carried out into the cell. So this permease is the one which increases the permeability of the cell to beta galactosidases. Another one is A gene, which encodes for transacetylases. So hence all these three gene products in lac operon are required for the metabolism of this lactose to take place. Let's understand this clearly. For example, an E. coli is grown in a medium containing glucose, which is a monosaccharide, Another E. coli is grown in a medium containing glucose and lactose. So, in the first Petri plate, this E. coli grown with the glucose medium will utilize the glucose, which is a monosaccharide, whereas the E. coli which is present in the second medium containing both glucose and lactose will prefer glucose rather than lactose because the lactose has to be broken down into glucose and galactose. So the E. coli will prefer to use glucose by itself. What happens when an E. coli grown in a lactose medium, the glucose, the lactose has to be converted or it has to be broken down into glucose and galactose, and the glucose will be utilized back by the E. coli. So, this lactose which is present has to be hydrolyzed first. So, the bacteria prefers to use glucose when it can. There are four situations possible. For example, we can take four plates, petri dishes. The first petri dishes, E. coli is grown in a medium containing glucose, whereas the lactose is absent. So what does the E. coli does? It prefers to utilize the glucose. So there is no need to produce beta glycosidase to break this lactose into glucose and galactose. So it directly takes the glucose. Whereas in the second case, for example, if the bacterium is grown in a petri dish containing glucose and lactose, here the E. coli prefers to utilize glucose rather than going and breaking down the lactose. So not necessary to produce this beta galactosidase. The third petri dish contains E. coli grown in a glucose absent condition and lactose also absent condition. At the time, there is no need to produce beta galactosidase because there is no glucose as well as there is no lactose. The fourth petri dish, the E. coli grown in the lactose conditions and the glucose is absent at that time, it is necessary for the E. coli to break down this lactose into glucose and galactose. So we require beta galactosidase enzyme to break down this lactose into glucose and galactose. The next one is very important. We need to understand this clearly. As already said, this I gene is the inducer gene which codes for repressor protein, and Z gene is the one which encodes for beta galactosidase, which we otherwise call it as beta gall, and Y encodes for permeases, and A encodes for transacetylases. So this lactose here acts as a substrate for the enzyme beta galactose. We all know that an enzyme and the substrate combines together forms an enzyme substrate complex. So when an enzyme is present, a substrate has to bind. So this lactose acts as a substrate for the enzyme beta galactosidase, and it is the one which is responsible for regulating on and off of the operon. So we call lactose as an inducer. The reason why it is mentioned here in the absence of inducer, in the absence of the presence of inducer is an inducer is the one lactose. It acts as a substrate for the enzyme beta galactosidase, and it is the one which is responsible for regulating switching on and off of this Lac operon. Next, we need to understand two different concepts here. The first one is when there is no lactose but there is glucose. So the E. coli directly utilizes the glucose and there is no need to break down this lactose into glucose and galactose. At the time, what happens is this orange color which is present here represent an operator region. So this I gene, which is a regulatory gene, an inhibitory gene, encodes for the repressor protein. It transcribes into a repressor mRNA and then translates into a repressor protein. This repressor protein always binds to the operator region of the Lac operon. When there is no lactose, what happens is this repressor goes and binds to the operator region. When this repressor binds to the operator region, it prevents the RNA polymerase to bind to the promoter region. Therefore, it prevents the transcription as well as the translation of all these three enzymes, and there is no beta galactosidase, no permeases, no transacetylase, because there is no need to synthesize all these three genes because there is no lactose to break down all these things. Next case is in the presence of inducer, that means in the presence of lactose. When a lactose is present, so the I gene, which is a inhibitory gene, transcribed into a repressor molecule, which is repressor mRNA, and then into a protein repressor. When an inducer, that is a lactose, is present, it goes and binds to the repressor, and when we already know that the repressor always binds to the operated regions, when this inducer, a lactose, goes and binds to the repressor, it inactivates the repressor so that the repressor, along with the lactose, which is binding onto the operator, is not active. Therefore, it leaves the site of the operon. So, therefore, this the RNA polymerases binds to the promoter region, initiating the transcription of the gene LAC-Z, Y, and A, finally resulting in beta galactosidase, permease, and transacetylases. This is an overview of what Lac Operon concept in the absence of inducer, which is a lactose, in the presence of inducer, which is lactose. The control of Lac Operon. First case is when lactose is absent. We already know when there is no lactose, what happens. The repressor protein always been synthesized, it always been present onto the operator site. So this repressor protein's role is to block the promoter site so that the RNA polymerase cannot bind to the promoter sites. Therefore, there won't be any transcription. So this takes place when there is no lactose. So there is no need to synthesize all these, transcribe and translate all these genes so that the lactose has to be broken down into glucose and galactose. The next case is when lactose is present. We have already seen when lactose is present what happens. A small amount of sugar allolactose is formed within the bacterial cell. So what happens is this is the yellow lactose, which is yellow in color. So this fits onto the repressor protein at another active site. One active site is present onto the operator region. Another active site is for this allolactose region. So this allolactose comes and bind to the another site of this repressor protein. So when it binds, it changes the conformational shape. So there is a conformational change. So it can no longer able to sit onto the operator site. So it moves away. So the RNA polymerase can now reach its promoter sites, and it prepares for the transcription of this LAC-Z, Y, and A to take place, resulting in the production of all the three enzymes. And this three enzyme, beta galactosidase, breaks down this lactose into glucose and galactose, and the permease transfers this glucose and galactose back into the cells, and the transacetylation takes place over there. The next one is when both glucose and lactose are present. What happens? We've already seen when lactose is present, when lactose is not present. But what happens when both are present? When both are present, the RNA polymerase can sit on the promoter site, but it remains unstable. For example, we already know the lactose, when it is present, it comes and binds to the repressor, and it removes the repressor from the operator site. When an RNA polymerase comes and binds to the promoter region, it is not stable and it keeps falling off. So the transcription of doesn't takes place for the Z, Y, and A genes. When glucose is absent and lactose is present, another protein is needed. When there is no glucose but there is lactose, what happens? So we need a protein called as an activator protein. It is the one which stabilizes the RNA polymerase to bind onto the promoter site. This works only when glucose is absent. So E. coli makes enzyme to metabolize other sugars in the absence of glucose. It's very easy. So when the lactose is present, it binds to the repressor molecule, removes the repressor from the operator sites so that the RNA polymerase, along with the activator protein, binds to the RNA polymerase, binds to the promoter region, It stabilizes the RNA polymerase, resulting in the transcription and the translation of all these three genes. So, this is the summary of what we have learned. So, there is a carbohydrate which represents two sugars, glucose and lactose, activator protein, repressor protein, RNA polymerase, and Lac Operon. So, the Lac Operon is possible in case when there is a lactose and when there is no glucose. So, first case is when there is both glucose and lactose. The activator protein is not bound to the DNA, so the repressor protein is lifted off onto the site, operator site. RNA polymerase is not stable, so it keeps falling off from the promoter site and there is no transcription takes place. The second case is when there is glucose but there is no lactose, so the activator protein is not bound to the DNA, whereas the repressor protein binds to the operator but it's being blocked by the repressor. The RNA polymerase is not able to bind to the promoter region and there is no transcription. The third case is when both are not present. So the activator protein is bound to the DNA, but the repressor protein is also bound to the DNA, but it's being blocked. The RNA polymerase is being blocked by the repressor and there is no transcription. Whereas the fourth case is the one when the lactose is present but there is no glucose, the activator protein is being able to bound to the DNA, the repressor protein is lifted off, it moves away from the operator sites, and the RNA polymerase sits onto the promoter site, bringing about the transcription of Lac operon, resulting in the production of three enzymes beta galactosidase, permease, and transacetylases. Thank you all once again.